And welcome back, Giving Tuesday at BTI. I'm your host for the day, Keith Hannon, and we are entering kind of the home stretch of the day. And I'm very honored to be joined by three members of our Emeritus faculty who are going to talk to us about their time at BTI. We just did the math, we ran the numbers, and we came up with over 130 years of combined BTI experience from these three outstanding researchers. So thank you all for joining us for this broadcast. I don't imagine there was a whole lot of live streaming when you first got to BTI, so thanks for humoring me with our experiments here of the communications nature. So let's first just, we have Bob, and Alan, but I'd like you to introduce yourselves and kind of when you tell us when you came to BTI and what you were kind of pursuing that brought you here.
Okay.
Or Yonkers in some cases, not necessarily right here.
Exactly. I'm Bob Granados. I started my adventure here at Boyce Thompson Institute on October 23rd, 1964, my first day of work at the Yonkers facility along the beautiful Hudson River. Great place. Nevertheless, I began my journey that led me here. Really, it started, I guess, in graduate school at the University of Wisconsin, where I was studying the transmission of plant diseases by insects. And in doing the literature research, as one should do as a graduate student, learning about the past and the current research, I realized right off the bat that two of the giants, two of the most recognized scientists doing the kind of work that I was excited to do were two individuals that had worked at Boyce Thompson Institute or currently was working at the Institute. The first of these was Louis Kunkel. Interesting individual, the first scientist ever hired by the Boyce Thompson Institute. He was the first one. And he worked on the transmission of Aspergillus diseases by a leafhopper vector. And did some really spectacular work at the Boyce Thompson Institute for 10 years before the Rockefeller Institute offered him a position he couldn't refuse, and he continued working there, eventually became a member of the National Academy of Sciences. Top-notch man. The second individual that happened to be there when I came to Yonkers facility was Karl Moreish, another outstanding scientists working in the same general field, working with the same disease, Aspergillus, and its transmission by leafhopper vectors. And he went on to win the Wolf Prize Award in agriculture, which at that time was considered a Nobel Prize in agriculture, an Israeli award that's given out yearly. So those were two very strong, compelling reasons why I came to the Institute. In addition, my PhD thesis was on the transmission of Aspergillus virus to plants. So I had those.
Seeing a trend.
You're seeing a trend, exactly. And that's how I happened to end up at the Boyce Thompson Institute Yonkers facility, and of course here in Ithaca, New York.
Great. Mm-hmm. Bob, how did you get to BTI?
Hi, I'm Bob Kohut, and I came to BTI because when I went to graduate school at Penn State, it was in plant pathology, and I was doing research on the effects of air pollution on plants. And among the places that some of the more interesting and pioneering research was being conducted was here at Boyce Thompson Institute. Through a interesting series of events that led me from Penn State to the University of Minnesota on a postdoc, and then out to an environmental consulting position in Fort Collins, Colorado, I became more and more aware of the fact that the kind of research that was needed to actually assess the effects of air pollution on plants had not yet been conducted. And that I made sort of a commitment to myself that if ever I had the chance to get into research, I was going to be interested in doing strictly applied air pollution work. And I ended up having the opportunity through, again, as I said, a interesting series of events that allowed me to apply for a position here. And the Institute had moved up to Ithaca by the time that I came, which was in 1980. And there was a group of researchers here that formed the environmental biology program at the time. And they were known not only in the U.S. but around the world for some of the research that they were doing. And so when I came here and interviewed and was offered an opportunity to join that group, it was, for me, a chance of a lifetime. It was the opportunity and a situation I could not turn down. So I was very glad to come here. That was in 1980. Did applied air pollution research, some really interesting and far-flung projects through 2004 when I retired. And since that time, over the summers, I've been fortunate enough to be able to do some consulting for the National Park Service on air pollution problems in the national parks. So that's kind of the nutshell story of my opportunity to come here and continue the work afterwards.
Great. And Alan?
Hey, my name is Alan Renwick. I came to the Institute in 1960 from Scotland. I had just got my degree in chemistry in Scotland, and I had been working at a horticultural research institute there. And they had a nice library which got the contributions of the Boyce Thompson Institute every month, I guess. And so I became quite interested in the idea of working over here. I wrote a letter to the Institute, not knowing who would answer it, but I got a letter back from the managing director, George McNew, at that time. He said, well, when you get to New York, why don't you just give us a call and we'll talk about it. So, so I arrived in New York on a Saturday night. On Monday morning, I called the Institute, and they told me to get on a train up to Yonkers, and I spoke to them for about an hour or so and started work the next day. So initially I was working with two chemists, Birchfield and Storrs, who were very much involved in writing a book on gas chromatography, which was new at that time. And they were looking at the biological applications of gas chromatography. And they wanted me to do some analytical work on looking for 2,4-D in milk, when cows had been in pastures which had been sprayed with 2,4-D. So this was kind of an interesting project, but they didn't have the analytical methods to do it. So a lot of what I did at the very beginning was to work out these methods. But in the meantime, I became very involved in gas chromatography and realized that I could use this for other things. And Birchfield and Storrs were also in charge of a analytical lab which served the whole institute, and so I got to use all the instruments that were needed, not only gas chromatography but spectrographs, UV visible spectrometry, and mass spectrometry. And so eventually I became in charge of this, the lab. And that led to later things in my career, which we might talk about. I got involved in the forestry program at the Institute, and this was probably the most exciting part of my life at the Institute.
Yeah, so that goes right into kind of my next question. You know, we talked with people all day here about from different levels. Some were kind of current grad students here, or later on in their scientific careers. Looking back on your own, what do you, when you think about what really, was your inspiration within your own research, what really drove your curiosity to kind of keep going with your work? What was that for you? And Alan, if you just want to keep going, that's fine.
Yeah, that's a difficult question, I think, because I came in at a lower level than most of the others. I had just my bachelor's degree. I went on to get my master's at City College while I was working at the Institute. And then when getting involved in the forestry program, this was a real challenge. They wanted someone who could look at the volatiles that were being given off from trees that were being attacked by bark beetles. And so I said, okay, gas chromatography is obviously the thing to use there. And I made a trip out to California where the Institute's lab was, and really got involved in the behavior of the insects and trying to figure out what was going on. And this is what really inspired me. We really need to know the mechanism of how these beetles can kill a healthy pine tree and go on. And the amount of damage that was being done by bark beetles at that time was 5 times that of forest fires. And so there was a real push to get something done about it. And so I was very happy to get involved in that. Mm-hmm.
Well, you know, as I said, when I came here, I had come out of an experience where I realized that there wasn't as much information about air pollution impacts of plants that one really needed to make an assessment of what the risk was. And the EPA had just recently been formed, and so they were requiring people to do environmental impact statements or environmental impact assessments. And so when I came here, the very first project I got involved in was one that was sponsored by the EPA, and what they wanted to know was, what is the impact of ambient levels of ozone and increasing levels of ozone on agricultural crops? What's the impact on yield, and how does that translate into an economic impact? So they had formed pulled together a group of institutions into what they called the National Crop Loss Assessment Network. And so there was a site here, there was one in North Carolina, there was one in Illinois, there was one in Oregon, there was one in Southern California. Each of the sites was going to do controlled ozone exposures in the field and examine the impacts on the physiology and the yield of important crops for that region. That information then was going to be sent to an economist working in Corvallis for EPA, and he was going to translate that information into what the present levels of impact were and what the economic consequences were and what would happen if ozone got worse. All this information eventually fed back into the regulatory setting process of EPA. So whatever the work was that was being done, it had an immediate use. It was within months of being peer-reviewed and published, was being used by the EPA in the standard setting process. So, I mean, I felt that we had a really important mission for us, and that particular project ran for 5 years. So there was a lot of work to be done. We had a field site here, just off campus, where we did all our controlled ozone exposures, and that information was flowing into the regulatory process in a very effective way. So, I mean, I really enjoyed that work, and I felt that, you know, we were doing something that was important for our country as well as for the science.
Great, thank you.
Well, when I got to Boyce Thompson Institute, I did continue some of the work that I had done as a graduate student on trying to understand the nature of the Aspergillus virus disease. I was interested in trying to identify the type of virus that very distinguished scientists had worked on it in previous years. It said it was a virus. We assumed it was a virus because it could pass through filters. They would normally hold back not a virus, but it was believed that the infectious agent could not pass through filters. Only a virus could. And anyway, I was very fortunate that soon after I got here, I discovered the power of the electron microscope. Electron microscopes in the early 60s were not very common at universities and research facilities. We had one of the first electron microscopes, a beautiful Siemens 1A, and it was available, and I was sent out to UC Berkeley to take a 3-week course on how to use electron microscopes, along with a lot of newcomers to the field. Came back, and I started to search for the Aspergillus virus. And at the same time that I was doing this, I was interested on the insect part, and I realized because of Karl Moreish's experience that the culture of these cells from a vector would be a very important tool to study the interaction between the insect cells of plant and the pathogen. So I was fortunate that Boyce-Thompson at that time had a number of scientists that came in from Japan who were very good cell culturists. They also were learning on the job, you might say, but they were extremely very gifted with their hands and sterile techniques, etc. And I learned along with them how to culture insect cells in flasks. And of course that opened up an activity that occupied a lot of my time for the next 50-some years here at the Boyce-Thompson Institute. The that's right. And the electron microscope came and went. The interesting story is that in the end, we found out it was not a virus at all. It was a new type of microorganism known as a mycoplasma. And we came within a year of publishing that data before we were beat to on the game by another team from Asia. Nevertheless, it was all a learning experience. Don't take everything for granted in science. Challenge it. There might be other answers there that might be the cause of some phenomenon that you were experiencing. So the route that I took in cell culture technology and working and developing cell lines became a very important tool. And actually the Boyce-Thompson Institute for a while, between the mid-60s and up till the early 80s, was a center of excellence in invertebrate cell culture technology. We were producing a lot of cell lines, and people were coming to train at the Boyce-Thompson Institute. And of course, it became a very important tool to study the replication of bona fide viruses in plants. That was a pretty exciting time. And the same thing with the electron microscope. We were actually able to see the virus and how it grew itself in insect cells, and that was exciting. And it was the type of research that set the stage for very interesting discoveries and progress in the field of insect-plant interactions.
So you spent so many years, all of you, doing this exceptional research, and then there came a point where you all decided to retire. In that time, have you continued to kind of watch the area of focus in how it has evolved? And has it evolved, or, in Alan's case, or have we defeated the beetle and that's it? I imagine there's still processes that continue to be explored from what we learned from that. So how have you watched in the time that you've kind of officially retired from your lab work? What have you seen in your areas in terms of how those areas have evolved?
Well, the area of that probably has consumed more of my time than I expected would have been the insect cell culture technology that arose from the work in the early 60s, mid-60s and early 70s. And by that time, the science was becoming quite mature. A lot of people were using the technology around the world. And we had developed a number of cell lines from insects, mainly Lepidoptera, butterflies and moths. They showed the unusual capacity to express or to produce high levels of certain proteins, recombinant proteins, as they were being called at the time of my retirement. They could be infected with a virus vector and with a recombinant virus, and this protein could be produced in high levels. Proteins that had a rather interesting and role in areas, health-related areas, where these proteins could be used as screens for anti-tumor activity, and just as tools in medical research in various different ways in serological type studies. In the meantime, as Paul Debbie mentioned briefly this morning, we did fortunately take this technology to the point of using a cell line that was developed here at Boyce Thompson Institute in Ithaca, New York, as a platform for the production of the human papillomavirus vaccine. And that was gratifying. I had been retired 9 years before that particular vaccine was given the government's blessings to be used in the United States commercially for vaccination. It had been already in 2007, 2 years priorly, being released for use around the world in developing countries, but our Food and Drug Administration was a little bit more cautious, and anyway, it was very gratifying to have something that of this type to occur, and you felt rather good that there was some important health application that our research led to. So those are things that happened to me after retirement.
Great.
Yeah.
Thanks. Bob? Certainly, I think we would all agree that air quality and air pollution is still rather topical right now.
It's sort of interesting because a lot of the research that I did here as part of an environmental biology program did feed into the regulatory process. And so the EPA has come up with air quality standards for ozone that have been pretty effective. And one of the interesting things about ozone is it's not released by any particular source. Ozone at the surface of the Earth, down in the troposphere, is produced by emissions of oxides of nitrogen and hydrocarbons. So that's why you have emission controls on cars, mainly to try to get ozone under control. And those have been pretty effective. Even though there are more cars on the highway today, ozone levels have pretty much stayed the same or gone down in a lot of places in the country. But one of the very interesting things about ozone that's come to the fore in the past, let's say 10 years, is that when you hear people talking about environmental insults from hydrofracking, it usually involves water quality. But you don't hear much about ozone, and yet ozone is produced every place there's been fracking in this country. There's been a significant increase in ambient levels of ozone. And whether that's in the Northeast in Pennsylvania, or whether it's in the West in Utah and Wyoming, ozone has increased significantly. It's part of the reason they're trying to control fugitive emissions from these sites. And a lot of the work that I did for the Park Service has been directed at looking at national parks where upwind from them there's a lot of fracking being conducted, and so the emissions from these sites undergo transformation in the atmosphere by photochemical reactions powered by sunlight, and in the West you got a lot of sunlight, so you get a lot of ozone produced, and it's moved into some of these parks, and the Park Service is interested in understanding what's happening there. So ozone has been around a long time from emissions from automobiles, and now it's shifted over to a great extent emissions from hydrofracking. So there's always going to be some interest in what's happening to plants. And there is a need for more research, but there are only probably 2, maybe 3 sites, universities, or other institutions in the United States today that are doing ozone research. When I was doing it, there were probably 6 or 7.
Wow.
But it's boiled down to just a couple, and there are only really probably 4 or 5 of us who do field assessments of ozone injury. So if it's an art, it's a dying art.
Well, I was going to say, so is that a sign of progress or a sign of we're not investing in it like we should?
It's both.
Okay.
It's a sign that we've learned a lot about what ozone can do to plants, and we understand the risk of it, but there's also a sense among institutions that previously would fund this research that we know enough about it that we don't have to keep doing it. So it's a tough situation, and there's really a lot of interesting work to be done. Since I retired, I've had 2 peer-reviewed publications from the work I've done in the Park Service. So there's the opportunity there to do some interesting things and to get the results published. So it's an interesting situation.
Great. And Alan?
Well, I've been very pleased to see the results of our bark beetle research being used now for control methods. We had a project going in California and also in southeast Texas, where there were huge outbreaks of southern pine beetles. And the way that they were controlling these was to cut down a tree as it was infested with beetles and then spray it with insecticide. What we found is that the bark beetles use a pheromone to mass attack the tree and overcome the resistance. The resistance of the tree is exudation of resin. But if there are enough beetles attacking at one time, they can overcome this. And what happened was we found that after identifying the pheromones that are involved, the predators and parasites are also responding to the pheromone. So they're tuning in on their prey. And so by cutting down the tree and spraying, they were the beetles were all safely under the bark. The predators were crawling around on the outside waiting for the new ones to emerge, and we were killing these. And so by cutting out the insecticide applications, it's made a huge difference. The other thing is using pheromone traps. Now that we know the chemicals involved, you can make sticky traps, and there are several companies now selling these. And this has worked very well in Idaho for the mountain pine beetle. And I think in general, just the forestry practices have changed. There's one thing that they're using in Texas, in addition to the pheromone, you can bait a tree in a certain area and pull in the beetles using the attractant, and then make a circle around it of another compound, which actually it's a compound that the beetles use to tell them that the tree is filled up, and it's produced by the males after they've mated, and it's a signal that the tree is occupied. You can use this to keep other beetles away. And so now there's a company producing this compound called verbinone, which can be hung from little sacks on a tree if you want to protect individual trees. So we've been very happy that this has gone. I should mention that when the forestry program was closed down, as we moved from Yonkers to Ithaca, I had to move into other things, more agricultural pests. And now I was interested in just the plant-insect interactions, how insects decide what plants to feed on and which ones to avoid. And this work has been continued by George Jander, who's doing wonderful work. He's taken it all to a whole new level, looking at the genetics and the evolution of resistance mechanisms in plants.
So Alan may have just answered my next question, which was going to be what research that's happening right now at BTI kind of has, would you say, you find yourself kind of most Interesting. Not to slight anybody's research at all, but is there one particular lab or area within BTI right now that you find yourself kind of really paying attention to and trying to learn more about?
Well, I think I just answered that. George Jander is doing this work, and yeah, I'm just fascinated by it. But I'll leave this to the next.
Well, I guess, I'm not a molecular biologist by any stretch. But I would say the work that's going on here right now that interests me the most is citrus greening.
Okay.
I've always felt strongly that, there are some really important and unique kinds of applied research that need to be done. And I guess that's what I like about that. I mean, they're going to the field, they're talking to growers, talking to orchard people. And that's sort of compatible with kind of thing that I did when I was here, the field work. So, yeah, I like that work a lot.
Great.
Well, there's a couple of areas that I'm following rather closely now. The one more recent one was a few years back when I heard about this greening disease project out of Michelle Heck's laboratory. And I was actually, to some extent, thinking back and saying, heck, a first scientist at the Boyce Thompson Institute was working in a similar area, transmission of diseases of plants by insects. Only Kunkel and Moreish and Granados, we were working with leafhoppers, which are first cousins of the psyllid. They're basically in the same order, and they're both suborders of Hemiptera. In any event, so there's a lot of things that they have in common. So, and then of course the work that I was doing here in the mid-'60s, along with Moreish, working on similar projects, and then seeing what's happening now. The science is, of course, like Bob said, it's very molecular, genetics, genomics, proteomics, you name it, and they're doing some very fine work. What is interesting is they're doing kind of work that we were doing back in the 60s, to some extent. We're looking at the pathway of this greening disease agent, a bacterium, as it is ingested into the alimentary tract, into the hemocoel, and back into the salivary glands. And we were doing things like that. We were actually dissecting insects and taking out salivary glands from these little microscopic leafhoppers, same size as the psyllids. And also we were analyzing the hemolymph and trying to determine the type of hemocytes were present, and et cetera. And trying to learn more about the defense mechanism that these cells might be, being used by the insect to protect them against some infection. So that has been very interesting to see that here it is now, 94 years since the hiring of Kunkel to now. We're still working in this topic, only using rather sophisticated molecular and genetics techniques. The other one is the continuation of the work that we did in cell culture research, and that is continuing. We have a laboratory headed by Gary Blizzard, where he is doing some really interesting work on engineering these insect cells so that they can more easily be infected with recombinant baculoviruses to produce, not only high levels of recombinant protein, or perhaps being used for some type of gene therapy research. And that work is moving along quite well, and I think there's a number of opportunities there, as we know from our intellectual property office in licensing. A lot of companies are still interested in this cell culture technology, and it's maturing. There's a great deal of interest, and I think that'll bode very well for the Boyce Thompson Institute in terms of a source of income with licensing down the road.
Great. Well, here's another question I have that's kind of, I don't know if this is going to be easy to answer or not. But I'm curious, if you think about what's happening in the world and some of the problems that we see and how science is trying to address some of those problems, whether it has been for a while or is just starting. Is there anything happening that if you had to start your career over, you would say, I'd like to work on that? You know, I'd be really interested in, even if you have to go through school again to understand something new, but I'd like to work on that problem, or I'd like to work on that issue, you know, that's being talked about right now. Anything come to mind for?
I'll pass this on to Bob while I think about that.
Well, I would just say that, you know, I basically consider myself pretty much a field biologist, and the reality today is that, you know, climate change is going to change a lot of things. And I don't think that we fully appreciate, nor have we really been told, how severe those impacts could be. I think there's a high probability that the impacts in natural ecosystems are going to be severe, and that agricultural ecosystems are also going to be impacted. I disparage that the fact is that we have not really come to grips with this. We've essentially wasted about 25 years of not dealing with the reality of what's happening. So, I mean, I think that there's a lot to be done there. The Institute is beginning to look around the edges of some of these things that involve agriculture and agricultural production. I would be personally very interested in looking at what's happening to natural ecosystems. One question that's always sort of interested me is, do plants adapt to changing levels of carbon dioxide? And if so, how do they do that? The perfect opportunity for plant ecology and plant molecular biology to get in bed together and take a look at that problem. So if I had to start over again, I guess I'd be kind of taking a look and try to figure out ways through collaborative research to take a look at CO2 as an evolutionary pressure on plant communities.
Great, certainly.
Yeah. I guess the one thing that I have become very interested in now, with all the forest fires going on, what is going to survive, and what are our future forests going to look like? And I think there are people in the Forest Service already working on this kind of thing. In fact, my daughter is doing some of it. But she's in Montana, and you know which, well, I guess we know already which species are most likely to germinate after a fire. And but I think there's an awful lot we don't know that needs to be looked at.
Great.
Actually, I've been thinking about this for a number of years, maybe 15 years or so, because, let me give you an example. The monarch butterfly, it's a very popular insect. It depends on the year, but you hear reports that the populations are declining, migrations are slowing up, the forests in Mexico are being cut down, and we're in serious trouble. This fantastic migration of this beautiful butterfly may come to an end, and the monarchs just might die out. It occurred to me a number of years back when here at Boyce Thompson Institute, we developed a cell line from the monarch butterfly, where you have living cells of the monarch butterfly growing in flasks. And we know we had the technology then, probably there's more sophisticated technology now of cryopreservation, because we can take these cells and put them in a special solution, and put them in liquid nitrogen. And 6 years later, we want living monarch cells. We can take them out, and they will start growing. Down the road, who knows when, if we were concerned whether it be the monarch or other animal types or other butterflies that are at the verge of going extinct because of climate change or others, and urbanization of the cities. There's a butterfly, a blue skipper that lives right at the edge of the landing strips at LA International, and their ecosystem is being drawn almost to nothing, and it's liable to go extinct. Well, we can develop cell lines. We can put them away in liquid nitrogen, and somewhere down the road, it sounds like science fiction, but the way science is moving, we may be able to construct or save that. The genome, of course, is going to be there, and reconfigure that butterfly again somehow in some way that we are not familiar with at this point. So it's another idea that I had. If I had a second chance, I might want to go into that area. And anyway, it's.
We can store the monarchs right next to Walt Disney. In the cryo freeze.
Oh, right next to Ted Williams.
That's right. Everyone else that's there.
Yeah.
No, well, thank you for sharing that, because I'm always really curious, how you all see the current state of things after giving so many years to kind of the current issues of your day versus what everyone is starting to deal with now, and how you might approach that. My last question before we go is, because it's Giving Tuesday, and it's always hard to talk about science without funding. They tend to go hand in hand, right? For better and for worse. So how have you, when you consider how you approached funding for your research when you were working, how do you compare that to what you see now? Do you feel this is a tougher time for scientists to get the funding they need? Or do you see there's opportunity in new funding streams, or is it a little bit of both? David Stern just kind of talked about how he thinks we need the government to do more, but it's also important for more private philanthropy to come forward to support science. So how do you kind of see the funding landscape right now compared to when you were going after your money?
Well, I think from my perspective, funding was somewhat difficult when I first started, and well into my early years here at Boyce Thompson. There was always a government money by virtue of the United States Department of Agriculture funding agency, NSF, NIH, the standards. I was not one of the early ones, but I was among the early ones that took money from industry. They were interested in some of the research I was doing, and there were no strings attached. I could publish. All they wanted to know is to what the progress I was making in the laboratory, and I think I like to see more of that happening, and I'm hearing that, in fact, there is an interest now in that kind of corporate money coming in. If we can work out a deal where they will not necessarily block our publication, they cannot, but be patient with us until we get a paper ready for press, and maybe hold it up for a couple of months if they want to patent something along with us, and then let us publish, or some kind of system such as that. And right now, but I think it could be long-lasting, or maybe it's just for a few years. There's a lot of money out there, a lot of money, and a lot of money that can be made by virtue of philanthropy. I was looking at this streaming this morning, and you can get excited listening to people like John Ramsey and Annie Cross in the green disease of citrus, and say, wow, that is fantastic. Young people, energetic, great speakers, and I would like to think there's some people with monies out there, if they see that show, they say, I might consider funding that. the Institute, because this is exciting work that needs to be done.
Well, when I came to the Institute in 1980, it was sort of the golden age of air pollution funding and research. So I was fortunate in two ways. First of all, coming here to a group that was well established and well known in research, and also coming here at a time when, compared to today, it was relatively easy to get research money. We had money from the EPA, as I mentioned earlier, to do this work on crop loss assessment. A little bit later, when I was here, we had money from a couple of agencies, EPA, the Forest Service, and also industry, the Electric Power Research Institute, to do a big study looking at the effects of ozone and acid rain on red spruce. That was a 5-year, $3.8 million research effort. So you can't lay hands on that kind of money too easily today. We got to work for the National Park Service up at Acadia National Park, and worked for Alaska Oil and Gas Association up in Prudhoe Bay, Alaska, looking at impacts of emissions on tundra vegetation. So the money was out there and the problems were out there, and so we had the opportunity to put those two things together into some really interesting pieces of research. Today, there's sort of a sense either that we know everything about this or that we don't care about it. A lot of the problems haven't gone away. I mentioned hydrofracking and ozone production. They're fracking all over North Dakota. They grow a lot of wheat there. They're fracking in Pennsylvania. There's a lot of forest there. They're fracking in the west, and there's a lot of riparian vegetation. Some of the more dry land stuff probably isn't ozone susceptible, but riparian sites are pretty unique and important. We don't understand what might be happening there. So I think that there is the need for more air pollution research today on a number of pollutants, but there's just not the funding or the interest in pursuing those kinds of problems today. So I was very fortunate in two ways, being here and having access to funding when it was needed.
Thank you. Alan?
Yeah, my early experience with funding is very similar to Bob Granata's here, that a lot of the money came from industry, especially in the forestry program. Once we started in California, the forest industry down in Texas saw the work that was being done, and they actually donated about 400 acres of forest to us and built a building for us. So the lab building was given to the Institute. Another source, also we had a field station in Idaho, which was supplied by Potlatch Forests, and they gave us the research money that we needed at that time. Another source of money that is a little unusual, the lab in California was built on an old gold mine, and belonged to Newmont, and Newmont donated it again to the Institute, about 800 acres. And all the rock that was brought up from the mining, it was in huge piles there. So someone came in with a rock crusher, and we were able to have them crush the rock, and for every truckload that they took off, the Institute got some money. So we had about $60,000 a year coming in from that. And we did some very selective logging over mature trees, and again about another $60,000 a year was coming in from that. Otherwise, we did get money from NSF. The key there was having a team of people, a forester, a good entomologist, an insect physiologist, and a chemistry, and together we were able to put together a pretty good story for NSF. That became more and more difficult as the years went on, though, and I think now, as I moved into the more agricultural systems, the only way I could get money was to collaborate with someone who was an ecologist. And the NSF and USDA do not have programs in chemical ecology, which was my field. So you really had to have partnerships or teams to be able to get funding. So the way I see it going now, philanthropy is probably the key. It's more and more difficult for people to get government grants.
Well, I will now allow you all to get out of the hot seats here. But thank you very much for joining me and sharing your insights and your experiences, because I think they're incredibly valuable and help us frame how we're addressing some of the current issues that we're facing and how we're conducting research right now. And I think there's always a lot to learn from how things have been done in the past if we're willing to pay attention to them. So thanks so much for sharing all of that. And I know I'm comfortable speaking on behalf of the Institute to say that it is really such a wonderful thing to kind of see you all around the hallways throughout the week. Maybe not every day, especially during golf season.
That's right.
But it really is great to kind of stop you and talk to you about different things and to get your experiences and your stories as someone who's trying to communicate the science. That's incredibly helpful. And so thank you so much. And we will be back in about 20 minutes for our final interview of the day with Delaney Sickler, who's going to talk about engaging the next generation of scientists. This is Giving Tuesday Live from the Boyce Thompson Institute, and we will be back in just a little bit.